Hello and a very warm welcome to my ninth video. In this video, the topic with which I am dealing is one of my favorites because the experiment is very easy and fun to do. Yes, you are right. This video has an experiment to explain how light intensity affects the rate of photosynthesis. Well, as you can see, this small tiny whiteboard of mine scribbled with so many stuffs, and therefore due to lack of space my next video will explain this experiments, observation and inference. But here my discussion is on the experimental setup. Okay, so this particular experiment is commonly known as Hydrilla bubbler experiment. Hydrilla bubbler experiment. For this we need a fresh hydrilla twig, as you can see here. A fresh hydrilla twig is needed for this experiment. Hydrilla is a freshwater hydrophyte or an aquatic plant and very much used in many of our plant physiology experiments. Then we need a slide, as you can see here, a beaker filled up with normal water, a beaker with water, thread, a small bubbler, thread and a small bubbler, and of course our very own Mr. Sun. So cut a small part of this hydrilla twig and very gently tie it to the slide with the help of a thread. Then attach the bubbler. Put this whole thing in the beaker having water up to its rim, that is up to its edge, as you can see in figure 3. And keep this whole thing first under bright light or bright sunlight for an hour. As you can see, there are two main conditions. First is bright light condition and the second one is diffuse light condition. So first keep this whole apparatus under bright light condition for an hour. And then later on keep this whole setup under low light condition, that is diffused sunlight, for an hour, as you can see in condition number B. You will see that in both the conditions bubbles are released from the bubbler. In this particular diagram, you can see that bubbles are released in both the conditions. Carefully note down the number of bubbles formed in both the conditions. Example, in condition number A, the number of bubbles are 19. If you count the number of bubbles in condition A, you will see that total there are 19 bubbles released. This number is not fixed. I have taken an arbitrary value. You can take any number, 10, 12, 20, anything. I have taken 19 here. And in condition number B, total 9 bubbles are released. So in my next video, my focus will be on the observation and inference that we have drawn or derived while doing this experiment. So till then, you all try to guess the reason for this difference in the number of bubbles for conditions A and B. And if you want to be sure about your guess or your answer, then you have to watch my next video. So please watch my next video. Thank you.